Hello, everyone. Thank you for attending this session. My name is Muhannad Al-Hamad, and I'll be talking about biology-guided neural network for species classification. So the problem we're targeting is classifying species given an input image. We focus on teleost fish classification, but this can be more widely applied, and we're trying to see if we can improve the performance of our deep learning models, if we can feed the phylogeny information, the tree of life of the fish into the model. So why is this important? Well, fisheries care about this for their feeding strategies. Ecologically, there are lots of surveys to make sure to measure the conservation efforts and prevent overfishing. And there are lots of scientific applications like studying interspecies behavior and other analyses that we care about. So how did early work do that? Well, experts used to try to extract certain measures like the length of the fish or certain landmarks, the distance between different fins, and use those measurements to do the classification, but that requires a lot of manual work and expertise knowledge. So more recently, machine learning has been very successful at image classification. That's both due to the rise in using GPU computational power and because there are now large image data sets that can be used for creating those models. So for example, ImageNet, which is one of the most important benchmarks, has millions of images from hundreds of classes such as chair, dog, and person. And those two developments really helped boosting the performance of machine learning. More specifically, it's the rise of a specific kind of machine learning called, or neural network called convolutional neural networks, or ConvNets. These ConvNets are specifically good at image classification. They have different layers. In the low layers, they learn low-level features like edges, and then they use those features to learn more complicated features and so forth. And in the high level, they can learn like complex features like patterns or skin texture, or even an eye. So how does this work? Well, machine learning needs data to train its model, to teach them. So we have pairs of image and species. We give the model an image of a fish, and we tell it this fish belongs to this species. And we keep feeding more examples, and the model is trying to fit all these examples, these pairs, by adjusting its parameters. So once the model learns, we evaluate it by showing it new images that it has not seen before. So we show it an image, and then we see if the model can, with a high probability, classify it correctly. So that's all good. And why do we think this is a challenge? Well, as I said, deep learning requires a lot of data to train. And that's not always possible in image classification. So for species classification, fish classification, we have found that wild fish collections from museums have very little data. So for the one we use, we have images from 575 species, 450 of them, 3/4, only have fewer than 30 images, which is not enough normally. And most of those images are, not most, many of those images are not usable because either the specimen is damaged, or it's missing parts like the tail, or it has some occlusion, self-occlusion, or some parts of the image are occluded, so it's not usable. So we can see that the scarcity of data is a real problem here. So we thought, how can we improve this? Well, we looked at the tree of life and or the phylogeny, and we saw that species that belong to the same genus tend to exhibit similar traits. So we can see here for this genus, these two fish look similarly more than their similarity to these fish, and vice versa. So we thought, can we use this similarity to our advantage? And we hypothesized that incorporating this phylogeny, this genus information, can help the model learn biologically consistent and relevant features, which would lead to better accuracy in the presence of limited data. So learning this genus features and species features would make it better. That's the hypothesis. And this is part of a trend that has been recently going in machine learning, which is called science-guided machine learning, where we try to use the domain knowledge in addition to the data to make the model better. So our model is called HGNN, Hierarchy Guided Neural Networks. And basically how it works is we have two convnets, one for the species, one for the genus. So when the genus branch learns the genus features, we take those features and we feed them for the species classification task. And like we hypothesized, we think that this hierarchy will help in the species classification task. So like any neural network, we need data, and we collected data from five different museums. Those are from the Great Lakes Invasives Network Project, which is called GLIN. And the total number of images is about 64,000 from 575 species. The images look like this. It has like a fish with a ruler for scale and some information on a label. We crop each of these components into its separate image. And for fish classification, we only use the fish itself with the information about the species and the genus. And for computational feasibility, we create two subsets, easy one that has 38 species from 11 genera. And we use this one to study the effect of the number of images per species, basically the effect of scarcity of data. And the hard one, which comes from all museums, whereas the easy one comes from a single museum, which is easier. And the hard one comes from 5 museums, 102 species, so it's a lot more complex and generally less data, less images per species. And we use 80% of these images for the training and 20% for the evaluation. So to make the model more robust, every time we feed the image into the model, we try to perturb it a little bit, change its direction or its intensity, color intensity, its position. And this helps the model learn features that are really relevant to the fish itself, rather than taking on nuances like the color of the background or the orientation of the fish. We also use something called transfer learning. So instead of starting the model from random initialization, we started from a model that was trained on ImageNet. And since ImageNet is a large data set, a model trained on it has already learned many low-level and intermediate-level features that could be useful for any image classification problem. So we start from it, and then we specialize into fish classification. We could have specialized into any other thing like flower classification or plane classification. But starting from this model will mean less training time and better performance in general. So how are our results? Well, if we compare the easy to the hard data set, we can see that looking at these box plots, we usually look at the central line, which refers to the median, because training a deep learning model is usually a stochastic process. So we try to do it multiple times, and we look at the median or the average. So we can see that for the easy model, our model that uses the phylogeny is very comparable to a vanilla model, which we call the black box. The median is almost the same. So there's not much of an advantage to using our model. But when we go to the hard data set, we can see that our model has a gain of about 1%. So we can see that our model does better when the data is complex and the amount of data is relatively low compared to the number of species. We also study the effect of data sparsity. So taking the easy data set, we try to take a version that has 100 images per species and another version that has 50 images per species. So as we saw, for 100 images, that's enough data to do the classification. So having the phylogeny did not really add much value. Whereas when we had 50 images, which is little data, we can see an improvement of about 3%. Now, what's more important is studying the robustness. So how do we do that? We use a tool called Saliency Maps. And Saliency Maps gives you a visualization of what is the model looking at when it's trying to classify the image. So here we can see that for this fish, the model is looking at the whiskers and some of the fins. For this fish, it's looking at the eye. Sometimes it's looking at something completely irrelevant. Like here, for example, it's looking at the label. So we know the model is not doing its job. It should be looking at the fish, not at the label. So it could also be a debugging tool. So robustness, how do we study it using the saliency map? Well, if we cover parts of the image, we can see if the model can still try to figure out the class based on other parts of the image. So for example, we start with this image. We can see that it's looking at the eye, and the classification is correct for both black box and HGNN. But if we cover the eye, we can see that the probability drops. It's still, the HGNN is still more sure about it, but the probability generally drops. And then if we cover more parts, we can see that black box is completely clueless. It doesn't know what, it doesn't classify it correctly, and not even within the right genus, which is the prefix here. Whereas HGNN still classifies this correctly, and even its second guess is still within the right genus. So we have shown that using the hierarchy has an advantage in more resiliency, better accuracy, and better generalization. We can also show that this concept can be generalizable beyond fish classification, can be used for other classification problems in biology. And finally, this work is part of, like, it can be even more generalized to using, instead of the phylogeny, we could use the ontology and to do fish segmentation, where the ontology makes sure that the segments, the relationship of the segments within each other to each other makes sense. That's all. Thank you very much.